If you deleted all the lipids from your body right now, your cells would literally dissolve, your nerves would short circuit, you'd lose the ability to regulate nearly every hormonal process, and you would, well, you'd cease to exist in a matter of seconds.
That is the scope of what we're doing today. Our mission is to decode the density of this textbook chapter, practically line by line. We're going to find out why olive oil is liquid, but butter is solid, and why that specific, that physical state matters for the flexibility of your blood vessels.
We're going to see how lipids act as electrical insulators in your nerves, which is just a fascinating piece of engineering.
And you mentioned before we started that we'll get into some very specific, somewhat surprising medical connections.
Oh, yeah. Like how the geometric shape of a single lipid molecule can determine if a premature baby can breathe properly. Or, and this is a wild one, how the cholera toxin manages to hijack a specific fat receptor in your gut to kill you.
It gets that specific, a single receptor.
It gets incredibly specific. The difference between health and disease so often comes down to the placement of a single double bond in a carbon chain. It's that precise.
I love that. Okay, let's unpack this. Section 1 involves, well, the text calls it biomedical importance, but it really starts with defining the undefinable.
That's a great way to put it.
In biology, we usually define things by what they're made of. Proteins are chains of amino acids. DNA is a chain of nucleotides. But the text starts by saying that lipids are a heterogeneous group. They don't have a shared chemical blueprint.
And this is the first hurdle in biochemistry, and it honestly confuses a lot of students. Lipids are the outcasts. They aren't defined by a common chemical structure, like a repeating backbone that you see in proteins or nucleic acids.
So what are they defined by?
They're defined by a physical property. Solubility.
The oil and vinegar rule?
It's slightly more technical, but yes, that's the core idea. The formal definition is, lipids are relatively insoluble in water, but soluble in nonpolar solvents.
What are nonpolar solvents?
The text specifically lists things like ether and chloroform. Think of them as oily or greasy liquids. So the definition is purely behavioral. If you throw a molecule into water and it refuses to mix, it just clumps together.
Right.
But then you throw it into, say, chloroform, and it dissolves completely.
Oh, yeah.
Congratulations, it's a lipid.
So we have this big, messy family of water haters. And because that definition is so broad, the text immediately outlines 4 massive biomedical roles they play. I want to break these down because they really set up the stakes for everything else we're going to discuss.
They do. They're the 4 pillars.
Okay, pillar number 1 is high energy value.
This is all about efficiency. And it's the one most people are kind of familiar with. Fat is the most dense energy currency the body has.
More than sugar.
Oh, much more. If you compare it to carbohydrates or protein, lipids hold significantly more calories per gram. You're looking at about 9 kilocalories per gram from fat versus only 4 for carbs or protein.
So it's more than double the energy density.
More than double. And think about that from an evolutionary standpoint. If survival is a game of packing a survival kit for a famine, lipids are how you pack light but carry heavy energy.
It's the difference between carrying a bag of feathers versus a brick of gold.
That is a perfect analogy. And there's another piece to it. When your body stores carbohydrates as glycogen, it has to store a lot of water along with it. But lipids are hydrophobic. They hate water, so they pack away neatly without all that extra water weight. It's pure, concentrated fuel.
Okay, rule number 2, structural insulators. The text mentions thermal insulation, subcutaneous fat keeping us warm, which makes sense, but I was more interested in the electrical side of things.
This is the myelin sheath, and it is a masterpiece of biological engineering. It's crucial. Your nerves communicate via electrical impulses, what we call depolarization waves or action potentials.
Like electricity flowing through a wire.
Exactly like that. But if those nerves were bare wires, the signal would just dissipate into the surrounding tissue. It would be incredibly slow and weak, if it got there at all.
So lipids are the rubber coating.
Precisely. Lipids, specifically sphingomyelin, which we'll get to later, form this wrapping around the nerve axon. This sheath allows the electrical signal to jump from node to node, traveling at incredibly high speeds. It's called saltatory conduction.
So when we hear about diseases like multiple sclerosis, what's happening there?
That is a demyelinating disease. For reasons we don't fully understand, the body's own immune system attacks and destroys that lipid layer. The insulation is stripped away.
The wire is exposed.
The wire is exposed. The electrical signal leaks out, it slows down, it gets garbled, and the nerve can't fire properly. The function, whether it's muscle control or vision or sensation, is lost because the lipid is gone.
Wow. Okay. Rule number 3 is transport. And this seems like a real engineering challenge. We just said lipids hate water, The blood is mostly water.
Yeah.
So how do you move fat around the body to get it from your gut to your cells without it just, clogging everything up like bacon grease in a cold pipe?
You build a submarine.
Yeah.
That's the best analogy. You have to package the cargo.
Mm-hmm.
You take the hydrosobic, that's the water-hating lipid part, and you put it on the inside. Then you wrap it in a shell of proteins and amphipathic lipids that like water on their outside.
So the outside of the submarine is water-friendly.
Exactly. The text calls these packages lipoproteins.
Mm-hmm.
We'll get into the different types like LDL and HDL, the bad and good, cholesterol, and later deep dives. But the principle is the same. It's a sophisticated trafficking system for moving grease through a watery highway.
And finally, number 4, nutrition.
This covers the things our bodies are just not equipped to manufacture. We can't make them from scratch.
Like what?
Primarily the essential fatty acids, which we'll discuss, and the fat-soluble vitamins. Vitamins A, D, E, and K. You can eat all the carrots you want for vitamin A, which is great.
Mm-hmm.
But if you don't have any fat in your digestive tract to dissolve it and carry it across the intestinal wall, it does you no good. You just flush it out. The fat is the vehicle that delivers the vitamin.
So already we see how this connects to obesity, diabetes, atherosclerosis. It's not just about aesthetics. It's about a potential mechanical failure in any one of these 4 fundamental systems.
Precisely. When lipid metabolism goes wrong, it's not just one thing. It's a cascade of potential failures across energy, structure, transport, and nutrition.
Correct.
Understanding lipids is understanding the machinery of these diseases.
Okay, let's try to get a handle on this diverse group. Let's move to section 2. Classification, or as I like to call it, organizing the chaos.
A very apt title.
The text breaks lipids down into simple, complex, derived, and neutral. Let's distinguish simple from complex first. What's the difference?
Simple lipids are, well, simpler. They are primarily esters of fatty acids combined with various alcohols. The 2 main actors here are fats and waxes.
Okay, so fats. Is there a chemical difference between a fat and an oil? People use those words differently.
Conversationally, yes. But biochemically, not really. It's purely a matter of melting point, a physical property. Oils are just fats that happen to be liquid at room temperature. Chemically, they are in the same bucket. Triacylglycerols.
So olive oil is just a melted fat, essentially.
You could say that. Waxes are slightly different. They're still esters, but they use a higher molecular weight monohydric alcohol instead of glycerol. They are much more structural, think beeswax, or the waxy coating on a leaf that prevents water loss. Much more rigid.
Okay, that's simple enough. Then we step up to complex lipids. This feels like where the biology gets really sophisticated.
This is where it gets very interesting, yeah. These are the workhorses of the cell membrane and signaling. You take that simple lipid structure and you add something extra to it.
An upgrade.
An upgrade. If you add a phosphoric acid residue, you get phospholipids. These are the primary fundamental building block of all cell membranes.
And the text breaks those down even further.
It does. It's based on what alcohol backbone they're built on. If the backbone is glycerol, it's a glycerolphospholipid. But if the backbone is a different, more complex alcohol called sphingosine, it's a sphingophospholipid.
Sphingosine. That sounds like a mythological creature.
It's actually named after the Sphinx. Because when it was first discovered, its function was a complete enigma to the researchers. They couldn't figure out what it was doing, so they named it after the riddle of the Sphinx.
That is fantastic. I love little details like that. Okay, so we have phospholipids, and then the text mentions glycolipids.
Right. And glyco is the prefix for sugar. So these are lipids that have a carbohydrate, a sugar molecule attached to them.
What do?
They almost always sit on the outside of cells with the sugar part sticking out. They act as ID tags. They can tell the immune system, hey, I'm part of the body, don't attack me.
Yeah.
Or they can act as receptors for signaling molecules, or even, as we'll see, for toxins.
Okay, so simple, complex. Then we have derived lipids.
This is more of a catch-all category. Derived lipids are just the breakdown products you get when you take simple or complex lipids and hydrolyze them, break them apart with water.
Yeah.
This includes things like the fatty acids themselves, glycerols, steroids, ketone bodies. It's the component parts.
And finally, neutral lipids. The text mentions acylglycerols and cholesterol here. Why the term neutral? What does that mean?
It refers to the electrical charge, or lack thereof. Phospholipids, for example, have a negatively charged phosphate group. They're polar.
Mm.
But acylglycerols, the main storage fats, and cholesterol are uncharged. They're neutral.
And why is that important?
It's crucial for storage.
Okay.
If your main energy reserve had a strong positive or negative charge, it would attract a huge amount of water and disrupt the cell's osmotic balance. Being neutral allows them to be packed away quietly in a corner inside a fat droplet in an adipose cell without disturbing the rest of the cellular environment. They're inert.
Okay, we've got the categories. A sort of family tree for lipids. Now let's zoom in on the fundamental building block of almost all of this, the fatty acid. Section 3.
The Lego brick.
The Lego brick. The text defines them as aliphatic carboxylic acids. Can you break that down for us?
Sure. Carboxylic acid just refers to the head group. It's a carbon double bonded to an oxygen and single bonded to another oxygen with a hydrogen is what makes it an acid. Aliphatic is chemist speak for a long, straight chain of carbon atoms bonded to hydrogen atoms.
So imagine a centipede where every segment is a carbon atom and it has a specific kind of head.
That's a pretty good visual, yeah.
And there is a rule mentioned here that I found strange, the even number rule.
It is strange until you see how the factory works. When you look at the fatty acids found in nature, in our bodies, they almost always have an even number of carbon atoms, 14, 16, 18. You rarely, if ever, see 15 or 17.
Why not?
That's because our enzymes synthesize them by adding 2 carbons at a time using a little molecule called acetyl-CoA as the donor. It's like building a Lego tower, but you only have the 2x2 blocks. You're always going to end up with an even-numbered height. You can't get to 15.
That's fascinating. The machinery dictates the final product size. Okay, that brings us to the nomenclature, the naming systems. The text dives deep into how we name these things, and it's surprisingly complex. You have the Genevan system, which seems to be the very formal chemical name.
Right, the super proper IUPAC name. If the chain is saturated, it ends in oic. So a 6-carbon chain is hexanoic acid. If it has double bonds, if it's unsaturated, it ends in enoic, like octadecanoic acid for an 18-carbon chain with a double bond.
But you almost never hear those names used.
Honestly, outside of a pure chemistry lab, we rarely use them. We use the common names like palmitic acid or oleic acid, and more importantly, we use the numbering systems because they tell us about the structure and the nutrition.
Okay, this is key. The delta system versus the omega system. I see this on supplement bottles constantly. Omega-3, what are we actually counting?
We are counting carbon atoms in the chain, but the direction you count from is what matters. Every fatty acid chain has a head and a tail.
The head is the acid part.
Correct. The head is the acid group, the carboxyl end. It's carbon number 1, and the other carbons are numbered sequentially from there, 2, 3, 4, and so on. The tail is the last carbon in the chain, the methyl group.
Okay, so the delta system counts from the head.
Correct. The Greek letter delta is used to signify a double bond, and the system counts from the carboxyl end. So if you see a fatty acid described as delta 91I, it means there is a double bond that starts at the 9th carbon, counting from the acid head. This is really useful for chemists who are synthesizing the molecule.
But the omega system, the one we see on food labels, counts from the other end, from the tail.
Exactly. Omega is the last letter of the Greek alphabet, so it represents the end. In this case, the methyl carbon end. So if you start at the very tip of the tail and count backward, 1, 2, 3, and you hit the first double bond, that is an omega-3 fatty acid.
And if the first double bond from the tail is at the 6th carbon?
It's an omega-6 fatty acid.
So physically, an omega-3 and an omega-6 just tells you how close the first double bond is to the end of the chain.
That's it. That's the entire definition. But that position, as we'll see, dictates what enzymes can grab onto it and what the body can turn it into. It changes the entire biological destiny of that molecule.
Let's talk about those bonds. This is section 4. Saturated versus unsaturated. This is probably the most common nutritional distinction we hear about fats. Saturated fat, 10 grams. What does that actually mean at a chemical level?
Saturated means the carbon chain is holding as much hydrogen as it possibly can. Every carbon atom in that chain has 4 hands for bonding. In the middle of the chain, 2 hands hold onto its carbon neighbors, and the other 2 hands are completely full, holding hydrogen atoms. It is saturated with hydrogen.
And visually, what does that molecule look like? What's its shape?
It's straight. A nice, linear, flexible rod.
Okay, versus unsaturated.
Unsaturated means that somewhere in the chain, 2 neighboring carbons decide to hold hands with each other twice. They form a double bond. To do that, each of them has to let go of one of their hydrogens. So they are unsaturated, they aren't full of hydrogen anymore. There's room for more.
And this is where it gets really interesting. This leads us to what the text calls geometric isomerism, the shape of the molecule. Cis versus trans.
This is arguably the most important concept for health in this entire chapter. It's a game changer. When you have that carbon-carbon double bond, it's rigid. It locks the molecule in place at that point. You can't rotate freely anymore.
And this creates two possibilities for the shape.
Two possibilities. It all depends on how the rest of the carbon chain is arranged around that rigid double bond. If the two parts of the carbon chain continue on the same side of the double bond, it's called a cis configuration.
And what does cis look like?
It puts a permanent kink in the chain, a sharp 120-degree bend. The molecule looks like a V shape or a boomerang. Most naturally occurring unsaturated fatty acids are in the cis form.
Okay, so cis means kinked. What's trans?
Trans means the two parts of the carbon chain continue on opposite sides of the double bond. The trans configuration essentially cancels out the bend. So even though it has a double bond, the molecule remains straight, very similar in shape to a saturated fat.
Why does straight versus bent matter so much to a cell? It seems like such a tiny detail.
It's all about packing. It's about geometry. Think of a stack of paper. The sheets are flat and straight. You can stack them incredibly tightly with very little space between them.
Right.
That is, saturated fat, which is straight, or trans fat, which is also straight. Because they can pack together so tightly, the forces between them are strong and they become solid at body temperature. They make membranes rigid and stiff. Think butter or lard.
And the cis fats, the bent ones.
They are bent. It's like trying to stack a big pile of crumpled paper or boomerangs. You can't do it. They push each other apart because of those kinks. This creates space or free volume between the molecules.
That makes them liquid.
That makes them liquid.
Yeah.
The forces between them are weaker. Olive oil is liquid at room temperature because it is full of these bent cis molecules that prevent the stack from solidifying. This is essential for the fluidity of our cell membranes.
So when the text links trans fats to cardiovascular disease and diabetes, is it because they are basically tricking the body?
That is exactly what is happening. Most trans fats in our diet are not natural. They're created by an industrial process called partial hydrogenation, which turns liquid oils into solid fats like margarine. They are synthetic imposters.
Oh, so?
They look like an unsaturated fat chemically. They have a double bond. But physically, they act like a saturated fat. They're straight. So they get incorporated into your cell membranes, but instead of creating fluidity like a cis fat should, they make the membrane stiff and dysfunctional. It compromises the mechanical integrity of the cell wall.
That is a terrifying image. Your cells literally becoming stiff because of these imposter fats.
It's a serious structural problem at the most fundamental level of biology.
Let's talk about the good guys then. Section 5. Essential fatty acids. The omega-3s and omega-6s we defined earlier, why are they called essential?
Simple reason? We have a design limitation. Humans, and all mammals actually, lack the specific enzymes, called the desaturases, that can create double bonds at the omega-3 or omega-6 positions.
So we can't count from the tail end.
We can't work on that end of the molecule. We have enzymes that can make a delta-9 double bond, but we can't put one in at the third or sixth carbon from the omega end. Since we can't make them, we must ingest them. We get them from plants or marine life. That's the definition of essential.
And the text highlights a divergence here, a sort of competition. Omega-6 with its parent molecule linoleic acid versus omega-3 with its parent alpha-linolenic acid, or ALA.
They compete for the exact same set of enzymes. It's a metabolic bottleneck. Both of them are starting materials that our body wants to elongate and desaturate further, turning them into longer, more potent signaling molecules called eicosanoids.
Eicosanoids, that includes things like prostaglandins and leukotrienes, right?
Exactly. And these are the molecules that control a huge amount of our local physiology, especially inflammation, blood clotting, and blood pressure.
So what's the difference in the signals produced from omega-6 versus omega-3?
This is the critical point. Generally speaking, the omega-6 pathway produces signals that are strongly pro-inflammatory. The prostaglandins and leukotrienes made from omega-6s tend to say attack, clot, swell up, which is necessary sometimes. You need an inflammatory response to fight infection.
But you don't want it on all the time.
You do not. The omega-3 pathway, on the other hand, produces signals that are much milder or, in many cases, actively anti-inflammatory and resolving. They are the calm down, resolve the inflammation, let's heal signals.
So if your diet has a ratio of, say, 20-to-1 in favor of omega-6, which the text says is pretty typical in the West with all the corn and soy and sunflower oils.
You are constantly, biochemically, pushing the pro-inflammatory pedal to the metal. You're flooding the pro-inflammatory pathway with raw material and starving the anti-inflammatory one. You're biochemically primed for a state of chronic, low-grade inflammation.
And this is why the text links long-chain omega-3s like EPA and DHA from fish oil to protection against things like cardiovascular disease, rheumatoid arthritis, dementia, even cancer.
Absolutely. You are fundamentally changing the signaling environment of your entire body just by altering the ratio of these two types of essential fats.
Okay, let's shift gears from signaling molecules back to storage. Section 6. Triacylglycerols. The text calls them tags.
Tags for short, yeah. These are the storage tanks. If free fatty acids are the cash in your wallet, triacylglycerols are the gold bars in the bank vault at Fort Knox.
What's their structure?
It's a simple, elegant design. It's a glycerol backbone, which is a 3-carbon alcohol, and it's holding onto 3 fatty acids via ester linkages.
And the text mentions some specific stereochemistry here that seemed a bit in the weeds. Carbons 1 and 3 aren't identical.
This is a detail for the biochemist, but it's important. It's about 3-dimensional specificity. Even though the glycerol molecule looks symmetrical on paper, in 3D space, enzymes can tell the difference between the top carbon and the bottom carbon.
So they have names.
They do. They're called the SN1 and SN3 positions, for stereo-specifically numbered. And it matters because specific enzymes load and unload fatty acids from these positions, and different fatty acids tend to attach to different spots. Usually you'll find a saturated fat at position 1 and an unsaturated one at position 2.
So the body organizes its storage very deliberately. It's not just a random pile of fats.
Nothing in biochemistry is random. It's all highly organized. But the main takeaway for tags is, this is what is in your adipose tissue. This is body fat.
So when you eat excess energy from carbs or protein or fat,
your body converts it to fatty acids, ships them to your fat cells, and assembles them into tags, which were then stuffed into a big lipid droplet. It's a brilliant survival mechanism. It protects the rest of the body from having high levels of free fatty acids floating around, which can actually be toxic to tissues like the heart and pancreas.
Okay, section 7. Now we get into the really complex stuff, the phospholipids. You called these the membrane makers earlier.
They are the absolute foundation of the cell. They're amphipathic, a word we'll define properly later, but it means they have a split personality. They have a polar head group containing phosphate that loves water, and 2 non-polar fatty acid tails that hate it.
And the structure is what allows them to form a barrier.
Yes, the bilayer. The most abundant one in our membranes, the text points out, is phosphatidylcholine, which is also known by its common name, lecithin.
Lecithin, I see that on food labels all the time, especially in things like chocolate bars.
It's used as an emulsifier in food, yeah. It helps mix fat and water together. But in the body, its main job is as a membrane builder. It's also a major reservoir of choline, which is important for making neurotransmitters.
The text highlights a very specific type of lecithin that fascinated me, the palmitoyl lecithin.
Ah, yes, the lung surfactant story. This is a perfect example of physics meeting biology.
Explain it for us.
Okay, so imagine the inside of your lungs, the little air sacs called alveoli. There are millions of them, and they're wet on the inside. Water has a very high surface tension. The molecules want to stick together.
Right.
So if you imagine deflating a tiny wet balloon, the sides will stick together because of that surface tension. If your alveoli stuck together like that every time you exhaled, you wouldn't have the strength to inflate them again. You'd suffocate.
So this specific lipid prevents that from happening.
The palmitoyl lecithin coats the inside of the alveoli, and it acts as a surfactant. It gets between the water molecules and breaks that surface tension. It acts like a molecular lubricant. The text notes that premature infants often haven't started producing enough of this specific lipid yet.
And that causes what they call respiratory distress syndrome.
Exactly. They struggle to breathe, not because their muscles are weak, but because they physically lack the specific molecular grease required to keep their air sacs from collapsing. It's a biochemical problem.
That's incredible. Let's move on to another one, phosphatidylserine.
Okay, so this one involves a process called apoptosis, which is programmed cell death. It's how the body gets rid of old or damaged cells in a clean, controlled way.
How does a lipid play a role in that?
So in a healthy cell, phosphatidylserine is kept exclusively on the inner leaflet of the cell membrane, facing the inside of the cell. But when a cell gets the signal that it's time to die, an enzyme called a flippase gets activated, and it flips this lipid to the outside of the cell membrane.
Like waving a white flag?
It's more like a dinner ball. That phosphatidylserine on the outer surface is a signal to the macrophages, the immune system's cleanup crew, that says, I am dying, come and eat me and recycle my parts. It's a molecular eat me signal.
A fat molecule is the signal for a cell to be eaten. That is unbelievable.
Then you have phosphatidylanosyl. This one is huge in cell signaling. It sits in the membrane and acts as a precursor to powerful second messengers. When a hormone binds to a receptor on the cell surface, an enzyme can cleave the head group off of phosphatidylanosyl, creating 2 new molecules that then go on to activate a whole cascade of processes inside the cell. It's a key part of the cellular switchboard.
And what about cardiolipin? Is that found in the heart?
It's named that because it was first isolated from heart muscle, but its real home is in the mitochondria, the power plants of the cell. And yes, it's critically important for heart function because the heart is packed with mitochondria.
What makes it unique?
It's a massive double phospholipids, like two phospholipids glued together. The text links decreased levels of cardiolipin to heart failure, and it's a specific genetic condition called Barth syndrome. It essentially acts like the glue holding the complex protein machinery of energy production together in the mitochondrial membrane. Without it, the power plant falls apart.
And one more in this group, plasmalogens.
These are unique. Instead of the typical ester bond at the first carbon of the glycerol backbone, they have an ether bond.
What's the significance of that?
An ether bond is chemically much more stable and resistant to attack by certain reactive oxygen species. The text notes they make up 10–30% of the phospholipids in brain and heart tissue. We don't fully understand them, but the leading theory is that they act as a built-in protective antioxidant shield for these vital tissues.
Protecting the brain. That's a good segue to Section 8. Sphingolipids and glycolipids. Brains and signals.
So now we swap the backbone. We're getting rid of glycerol. Now the platform we're building on is sphingosine.
And the simplest one is ceramide.
Ceramide is just sphingosine with a fatty acid attached. You might see that in expensive skin creams. It's a key regulator of the skin's water permeability barrier. But in a biological sense, it's the parent compound for all the more complex sphingolipids.
Like sphingomyelin.
Right. Take ceramide and add a phosphocholine head group, and you get sphingomyelin. Exactly. This is the other major lipid component of the myelin sheath, the insulation for nerves we talked about. It's incredibly important for brain function. But it's also a key component of what are called lipid rafts.
What is a lipid raft? That sounds strange.
Imagine the cell membrane is a fluid dynamic ocean. A lipid raft is like a small, floating island of more rigid, tightly packed lipids, like sphingomyelin and cholesterol floating in that ocean.
And what happens on this island?
This is where a lot of signaling happens. The raft gathers specific receptors and signaling proteins together in one place, making the process much more efficient. It's like having a dedicated conference room on the surface of the cell.
So it's an organizing principle.
A major one.
And then we have the glycolipids, the ones with sugar, specifically the glycosphingolipids.
Right. So now we're taking that ceramide base and adding carbohydrates to it. These are almost exclusively found in the outer leaflet of the plasma membrane, forming what's called the glycocalyx, a sticky, sugary coating on the cell.
The text mentions gangliocytes here, and it points out a killer detail regarding the disease cholera.
Ah, yes. The GM1 ganglioside. It's a very complex glycolipid found on the surface of your intestinal cells. It has a normal physiological function related to cell recognition. But unfortunately for humans, it happens to be the perfect landing pad for the cholera toxin.
So the bacteria is specifically targeting this one type of fat.
The toxin has evolved over millennia to recognize the specific shape of the sugar head group on the GM1 ganglioside. It binds to it with incredible affinity, which allows the toxin to enter the cell, where it then triggers a cascade that causes massive, life-threatening water loss.
It's a tragic case of biology being hijacked. The lipid is there for a normal function, but the bacteria exploits it as a backdoor into the cell.
Exactly. It's a molecular Trojan horse.
We can't do a deep dive on lipids without talking about the most famous or maybe infamous one. Section 9, steroids and cholesterol.
The word cholesterol triggers so much fear and anxiety. But if you look at the text, the first thing it does is call it a precursor of many biologically important steroids.
It's a building block, not a villain.
It is absolutely vital. First, cholesterol itself is amphipathic. It slots into our cell membranes and regulates their fluidity. Think of it as a fluidity buffer.
What do you mean by buffer?
Well, if the membrane gets too hot and starts to get too fluid, too runny, cholesterol's rigid ring structure holds it together. But if it gets too cold and the fats start to solidify, cholesterol gets in the way of the packing and keeps the membrane fluid. It works at both high and low temperatures to maintain perfect integrity.
And it's the parent of all our steroid hormones.
All of them. All the sex hormones. Testosterone, estrogen, progesterone. Also cortisol, the stress hormone, and aldosterone, which controls blood pressure and salt balance. They're all carved out of a cholesterol molecule by different enzymes.
And vitamin D.
Yes. The text mentions a molecule called 7-dehydrocholesterol, which is present in our skin. When UV light from the sun hits it, it cracks one of the steroid rings open, and that's the first step in converting it to active vitamin D. No cholesterol, no vitamin D synthesis in the skin.
The text also mentions ergosterol.
That's the equivalent molecule found in plants and fungi. When it's irradiated, it becomes vitamin D2,
And what about bile acids?
also made from cholesterol in the liver. And you need bile acids to emulsify and digest the fats you eat. So cholesterol is actually required to help you absorb and digest other lipids. It's a beautiful self-regulating cycle.
So to be clear, high levels of a certain type of cholesterol in the blood is a risk factor, but no cholesterol in the body is instant death.
Correct. It's about balance, transport, and regulation, not about the molecule itself being inherently evil.
The text also lists polyprenoids. They aren't steroids, but they are related.
They share a common 5-carbon building block called isoprene. They aren't steroids, but they're built using similar biochemical pathways. The absolute star of this group is a molecule called ubiquinone.
I know that one, coenzyme Q10.
That's the one. It's a lipid-soluble molecule that lives in the inner mitochondrial membrane, and its job is to shuffle electrons along the electron transport chain to make ATP, our body's energy currency. Again, a lipid-like molecule is absolutely central to us generating power.
Okay, section 10. This feels like the scary part of the movie. Lipid peroxidation.
This is the dark side of lipids. This is why fats go bad. If you leave butter out on the counter for too long, it goes rancid. That chemical process happens inside your body, too.
The text breaks it down into 3 stages: initiation, propagation, and termination. It sounds like a military operation.
It kind of is a destructive one. Initiation is when a free radical, which is a highly reactive molecule with an unpaired electron, desperately looking to steal an electron from somewhere else, attacks a fatty acid.
And it attacks a specific kind of fatty acid.
Yes, and this is the tragic irony. It preferentially attacks the polyunsaturated fatty acids, the healthy omega-3s and omega-6s we just talked about.
Why are they the most vulnerable?
Because those double bonds create weak points in the carbon-hydrogen bonds next to them. They're easier to break. So the free radical comes along, steals a hydrogen atom electron and now the fatty acid itself becomes a radical.
And that's the second stage, propagation.
Right. It's a chain reaction. That new fatty acid radical is unstable, so it reacts with oxygen to become a peroxyl radical, which then attacks its neighboring fatty acid to steal a hydrogen to fix itself. But in doing so, it turns its neighbor into a radical.
So it's a domino effect.
A domino effect of destruction ripping through the cell membrane, destroying hundreds or thousands of lipids from one initial hit.
And the consequences of this?
The text lists them. Damage to tissues, cancer, inflammation, atherosclerosis, aging. The process doesn't just damage lipids, it creates toxic byproducts that damage proteins, DNA, everything in their path. It's molecular shrapnel.
So how do we stop it? The third stage, termination.
We need a hero. We need an antioxidant.
Vitamin E?
Vitamin E, or alpha-tocopherol, is the body's premier lipid-soluble antioxidant. It lives inside the membranes, right where this damage is happening. When a peroxyl radical comes along, vitamin E steps in front of the chain reaction and says, take my electron instead.
So it sacrifices itself.
It sacrifices itself. It's a chain-breaking antioxidant. It neutralizes the radical and stops the dominoes from falling.
So vitamin E is literally a shield for your cell membranes.
It is the primary shield, yes, along with a whole network of enzymes like superoxide dismutase and catalase that deal with the initial free radicals. It's a constant battle inside our cells between oxidation, which is like rusting, and antioxidation.
The text mentions a molecule called malondialdehyde as a marker for this process.
Yes, when a polyunsaturated fatty acid with 3 or more double bonds gets destroyed by this process, it can break apart, and one of the pieces of debris is malondialdehyde. Doctors can actually measure the levels of this in your blood as an indicator of how much oxidative stress and lipid peroxidation is happening in your body.
So it's a measure of internal rancidity.
That's a grim but accurate way to put it.
Okay, let's wrap up with the final section. Section 11. Amphipathic lipids. We've used this word a few times now. Let's give it a formal definition.
It's the key to how life organizes itself in water. Amphipathic comes from Greek roots, meaning both loving. It describes a molecule that has a split personality. One part is hydrophilic or water-loving. The other part is hydrophobic or water-hating.
And pretty much all the structural lipids we've talked about, phospholipids, cholesterol, sphingolipids, are amphipathic.
They are. And this dual nature forces them to self-assemble when you put them in water. They don't just float around randomly. They organize.
And this leads to specific formations. The first one is a micelle.
A micelle, right. Think of a tiny sphere where all the hydrophobic tails point inwards, hiding from the water, creating a greasy core. And all the hydrophilic heads point outward, happily interacting with the water.
And what's the purpose of that?
This is how soap works. The greasy core traps dirt and oil. But more importantly, this is critical for digestion. Bile acids form the cells in your intestine to trap the fats you've eaten so that enzymes can break them down.
Then we have liposomes.
These are a bit more complex. They are spheres made of a full lipid bilayer, so they have a watery core inside, enclosed by the membrane.
And this has medical applications.
Huge ones. We can use these as drug delivery vehicles. You can put a powerful cancer drug, for instance, inside the watery core of a liposome. The body just sees a harmless little ball of fat, so it can travel through the bloodstream to the tumor, where it releases its payload. It's a form of nanotechnology.
And finally, the most important structure of all, the lipid bilayer.
This is the fundamental structure of all biological membranes. It's two sheets of phospholipids arranged tail to tail. The hydrophobic tails form the core of the membrane, and the hydrophilic heads face the watery environment on both the inside and the outside of the cell.
It creates a wall, a barrier that separates inside from outside.
And that separation, that boundary, is the very definition of a cell. Without that lipid bilayer, we are just a disorganized soup of chemicals. With it, we become a living, compartmentalized, highly organized organism.
This has been, honestly, it's been mind-blowing. I will never look at a bottle of olive oil or a stick of butter the same way again.
It's a surprisingly complex and elegant world, isn't it?
So let's try to summarize this journey. We went from defining lipids not by what they are, but by what they dissolve in. Their hatred of water.
Right.
We looked at the dense, efficient energy banking of triacylglycerols. We saw the structural genius of phospholipids and sphingolipids making membranes, insulating nerves, and even signaling for a cell to die.
We saw how the simple geometric shape of a molecule cis versus trans can determine the health of your arteries and the fluidity of your cells. And we saw the constant battle between free radicals trying to cause chaos and antioxidants like vitamin E standing guard.
And we rehabilitated cholesterol. A little bit.
It deserved it, just a little bit. It's a hardworking molecule.
So here's my final thought for you, the listener. We spend so much time obsessing over low-fat or high-fat diets, counting grams. But what this deep dive into the source material shows us is that the quality, the type, and the structure of the lipid is what really matters. It's biochemistry, not just calories.
Absolutely. And if I can leave you with a provocation to mull over, think about that double bond. Just a tiny shift in electrons, a little 120-degree kink in a long carbon chain, is the difference between a fluid, healthy cell membrane that can receive signals and a rigid, dysfunctional one that can't. Life, health, and disease hang in the balance of these microscopic geometries.
The secret life of lipids, indeed. Thanks for listening to this deep dive.
Stay curious.